Okay, I might as well get started. I know a few people might trickle in within the next minute or two. So I want to introduce myself. I'm Jessica Zebracki, one of the PGY1s here, and I wanted to go over my GROW presentation, which is pharmacogenomics, and more specifically in relation to oncology and its applications in electronic health records. I also wanted to give some consideration to my content expert, Mike Fallon, who helped coordinate all the content and make sure that everything was appropriately resourced and validated. I also wanted to give a disclosure, as I'm going to be talking about EHR applicability, that this presentation is intended only for internal use and no distribution of any EPIC-related materials is allowed outside of UW Health. So for today's grill, we're going to be covering three main points. The first of which is the introductory background to what is pharmacogenomics. So the identification of the types of genetic mutations, pharmacogenomic testing, and genomic sequencing methodologies. And then what we're going to do is we're going to build on that and apply it with the selection of appropriate pharmacogenomic resources based on the intended clinical application, and that's in the scope of oncology medications. And then lastly, we're going to discuss future applications within the electronic health record for pharmacists to utilize this pharmacogenomic data and actually apply it to patient care. So a brief background into the magnitude of genetics research, because I feel like as pharmacists, we don't talk about this as much. So to start off, I think a lot of us are aware of the Large Human Genome Project, the massive undertaking where this production of resources through federal grants and whatnot ended up costing $2.3 billion to sequence the entire human genome. And currently, we have able to bring that down to less than $1,000 to sequence a single human genome, which is a massive undertaking. There are currently 123 targetable biomarkers, and that's out of 483 total listed drugs within the FDA database. And typically, well, you'll find in a lot of these patients who get genomic testing, you'll find at least one atypical drug response based on the genetic variants. Testing prices do vary, and we'll go into the types of tests, but they are a wide range, what makes it hard to communicate the cost to patients. So at the most basic levels, most simplest variants that are identified can be as low as $300, but once we're talking about whole genome sequencing, which I'll explain in a short while, that can reach up to $5,000. And as a part of one of Obama's initiatives in 2015, we have a precision medicine initiative, which allows for the Department of Health and Human Services, Department of Veteran Affairs, and Department of Defense to invest in new research, technologies, and policies that help patients get individualized care for genetic testing. So there are actual US dollars that are dedicated to advancing genetics research. Now, before we get into this further, we should first talk about the types of mutations, and they can be categorized in two basic structures. One is germline and the other is somatic. So germline mutations are detectable mutations within the germ cells, so the sperm and ova that you see on the left, and that affects all cells within the body. The somatic mutations, on the other hand, are acquired mutations. So those typically only affect a portion of the body. Those don't get passed on to offspring typically. And the most common presentation that we see this in is mutations that are present in a tumor, not throughout the body. So expanding upon germline mutations, we have these hereditary cancer markers that some of us might be familiar with. So I can first explain the BRCA1 and 2 that are present in breast and ovarian cancers, certain ones. So these are present in 12% or rather, breast cancer is present in 12% of the general population, and the risk for malignancy increases by around 50 to 70% if these mutations are present. And in terms of BRCA1 in particular, the patient is more likely to present as what we call triple negative. So that leads to poor prognosis. That means that they have less markers that we can target because they're estrogen receptor negative, progesterone receptor negative, and HER2 negative. So having this information available for our patients helps us guide treatment. The same can be said about VHL and renal cell carcinoma, though the data supporting prognostic and predictive markers is somewhat unclear. We've seen that in some cases, in one Japanese study, we've seen certain VHL mutations indicate improved outcomes with stage 3 cancer, but we've also seen cases linked to a poor prognostic factor with certain VHL alterations. So this shows that there's definitely a lot that needs to be done to help us determine what mutations are indicative of certain prognostic factors. Then with germline mutations, we also take into account gene and drug interactions. So those are drugs with pharmacokinetic interactions with drug metabolism, drugs that might have a narrow therapeutic window, and certainly we'll think of our CYP enzymes, so the enzymatic function alterations with these germline mutations that can vary how, again, with the pharmacokinetics, how the drug is processed and eliminated. So here are some of our germline examples. As you can see, for the most part, these impact the toxicity of the drug as well as efficacy, though as a whole, we also look at, again, prognostic factors and predictive markers as well. Two that I'd like to highlight here are imatinib, which has a pharmacogenomic marker BIM, and methotrexate, which has the SCL01B1 as an indicator of toxicity. So with BIM, we found that this typically can have a sorry about that. We found that typically what it can do is it can show resistance pathways in BCL2 overexpression. So this pharmacogenomic marker helps us determine how well the patient can be impacted by treatment. And with methotrexate, we've seen that presence of a specific mutation within SLC0101B1 is indicative of a decreased drug clearance and increased methotrexate toxicity, mainly hepatotoxicity. So now moving on to somatic mutations. There's multiple types, so I'll go into a few main ones to highlight, and those are often amplification mutations. So those are increased gene copies that lead to increased transcription. Example here is HER2 overexpression due to amplification that is roughly present in 15% breast cancers. And then over here, we see point mutations. And that's where a single nucleotide variant in the DNA sequence causes an impact in the amount of protein that is present. So as you see here, the silent mutation has one amino acid that doesn't modify the protein that is created, so there's no change. With missense mutation, we have one change in the amino acid, which changes the protein, but only doing so in a very small sense. And so then we get into a bigger disruption with our nonsense mutations, where there's a full stop. So once we get a codon, which has an amino acid change to prevent further protein development, that results in major changes and shortens the protein. And those are mutations that typically cause the biggest impact. Frameshift mutations are additions to the nucleotides. So they can also cause a disruption, but instead of stopping protein synthesis, it might disrupt what types of proteins are being built. So as you can tell, if you insert a single nucleotide that's different, it can drastically alter the types of proteins that are built after, at which point that frameshift mutation occurs. An example of this is in BRCA1, you can see some frameshift mutations present. With splicing, this is less common, but it's also a nucleotide variation that alters how the RNA splices, and that's an addition or deletion of large portions of the genetic code that can also impact protein synthesis. Some somatic examples that we have here are mainly impacting the efficacy of the drug. And one to highlight in particular is crizotinib with a pharmacogenomic of ALK. So as I've mentioned before, this is an indicator for our providers and as pharmacists for us to be able to identify if a patient is ALK positive, that can indicate that this crizotinib drug might have improved efficacy. But as a whole, with all of these germline and somatic mutations, they can be used for prognostication, impacting or identifying how the drug may be metabolized, risk of disease development, and treatment efficacy. Moving on from our mutation types, I wanted to go into a little bit about how we sequence. So we started out in its infancy with first-generation sequencing methods, one of which is a very rough and dirty way of doing it. It's called Sanger sequencing. So what happens is you have these building blocks called deoxynucleotides or DNTPs. And you have a mixture of these, as you see in the middle here, that have fluorescent markers that are used by DNA polymerase for DNA synthesis. So they pick up on these markers and have a certain mixture of ddNTPs, which stop DNA polymerase. So these ddNTPs leave a specific color that's unique to each nucleotide when it stops and creates differing fragments. And so in step 2, as you see, the size separation by capillary gel electrophoresis will then help us see the largest to the smallest fragments. And through that fluorescence, you're able to scale back and identify which nucleotide, based on the fluorescent color, is identified at which space in the DNA sequence. Now, the problem with this is that it's a fast test, but it's only feasible for sequencing around less than 700 base pairs. So this can't be impacted on a larger scale. This can mainly only be utilized in smaller sections of your DNA. Next comes the second and third generations after Sanger sequencing, which is called next generation. So these were new processes that optimized the time and cost of sequencing. And they were able to use millions of reactions in parallel without the need for this gel electrophoresis. So we're able to save a lot of time, and we're able to make sequencing for amounts of DNA that were significantly longer than 700 base pairs. We also have hotspot or targeted sequencing, and that identifies specific genes to sequence. So it's even less expensive than next generation sequencing because similar to Sanger, we can pinpoint a small amount of base pairs, and the less amount of base pairs you need to test, the less resources you need to use and the more cost effective it becomes. But as a result of that, it's also less broad, so you might potentially be missing some variants or mutations that would otherwise be flagging as less hot. And then we have these all-encompassing whole exome and whole genome sequencing tests. So whole genome tests sequence all variants, and they're all-encompassing, but they come with the caveat that it's a large amount of information, requires a large amount of storage to keep this data in a computer database. And whole exome sequencing shows protein coding genes and is a slightly less data intensive way of still identifying a majority of the variants, around 80 to 85%. And those try to optimize our resources in order to highlight the ones that are most likely to have a disease link and ones that are actionable for us from either a prognostication or a drug target standpoint. Now, for your reference, I also included a list of various next-generation sequencing methods. There's a multitude of them, and every company has tried to find their own unique spin on it. One of which I found most interesting to highlight how complex these can be is ion torrent sequencing. So these use semiconductor chips to sequence DNA. And to put into context how small this scale is, the DNA goes into specific wells that are only 3 micrometers in diameter. That is smaller than I can possibly imagine. And these wells are running in parallel, hundreds of them. So what they end up doing is similar to Sanger sequencing. You cut the DNA into several fragments, and then they place them into these beads that you see in the pink here. Now, these beads are covered with millions of single-stranded DNA fragments, and these beads float into the wells located in the chip, and they're covered in a solution that contains the nucleotide. So again, referencing the Sanger sequencing, they utilize these stop nucleotides, but instead of having a fluorescence, we use pH monitors. So in the bottom here, it has a sensor that can identify voltage changes with pH, and each unique nucleotide corresponds to a unique voltage change. So this is a way to run multiple tests in parallel, and it's able to sequence these genomes relatively quickly in comparison to our first generation. Now that we've gone through a little bit of the background, I want to reach our first assessment question. So a single nucleotide variant within the MSH2 gene is found to be linked to hereditary non-polyposis colorectal cancer and breast carcinoma. Instead of encoding arginine, a base pair in the DNA is replaced and the sequencing terminates. So that is a stop codon. Which of these most resembles the appropriate mutation? Point, amplification, frame shift, or nonsense? Perfect. I've seen a lot of D's in the chat. So that's correct. That's a nonsense mutation because it ends up terminating the entire sequence. And as I've mentioned, that has a very dramatic effect because it fully stops the further progression of all the other proteins downstream from that DNA from being synthesized. Now we have a lot of different types of pharmacogenomic testing available. There's a lot of companies that specialize in solely getting this DNA organized for patients in a patient-friendly manner, as well as a physician-friendly manner. So those are, some of the few are Caris, Guardant, NeoGenomics, Strata, Tempus, and Foundation One. To highlight a few others here, just to give an example of how they utilize these tests and what it looks like for patients as well as providers, here is Invitae, which offers single gene, multi-gene, and whole exome sequencing. So they analyze the 25 most common genes that impact drug metabolism. And you can see those in the bottom left, that primary panel that they utilize. And you're still allowed to single out other variants, but typically I've seen that they will use this primary panel more commonly in patients. And to the right, you can see how the diagnostic testing results come out should a patient end up resulting positive in one of these. Another company is called Prevention Genetics. So what they do is they offer both standardized genetic panels and gene-specific testing, similar to Invitae. And a lot of these offer both of those, mainly from a cost perspective. As I've mentioned before, the smaller tests are running in the range of hundreds versus thousands. So it might be more advantageous to a patient if they know that they're looking for a single variant or concerned for a single variant to have smaller tests available. And to the right and to the bottom here, you can also see examples of how the information is read out to patients and providers if there is a specific variant targeted. And it also provides the allele frequency as well as links to other databases so that providers are able to look into the data further. And lastly, within the interpretation section, you're able to see if there's any actionable interventions as a result of the information that they provide. Strata is another panel that is more specific. It has 437 genes. It's a pan-solid tumor next-generation sequencing assay. And as you can tell, it targets a lot of somatic mutations that can be present in a lot of our oncology patients. Some of the ones highlighted here are KIT, MET, KRAS, NRAS, and the list goes on. So this is a very wide net that we can cast for patients. Some of the differences that we need to consider when looking into NGS panels is coverage. I've hinted at this throughout this presentation because I really want to drive down the point that it's important to have that in the context of when you're having these discussions with your providers. If they end up deciding to implement a sequencing test for the patient. So Sanger sequencing and targeted tests can range from the 300 to 700 range, and then whole genome or exome sequencing is at least 1,500 to 2,000 plus from the examples I've shown you. There's also short read lengths of roughly 350 base pairs. So it's a barrier that we're not able to do a larger amount at this point. It's harder to get to more than 700 base pairs at one time, that we can run multiple tests in parallel. So we've tried to find a way to work around that barrier. And lastly, we're unable to sequence without an initial library creation from the sample DNA. So what they have to do is they take the DNA and they place specific markers within the DNA sample so that within their computer database, they're able to catalog where that DNA sequence or fragment is located. And that's because we only have short read lengths available. So that is a logistical barrier that also impacts why some of these sequencing tests are so expensive, especially the whole genome sequencing tests. So now that we've talked about, excuse me, so now that we've talked about all the guidance that's out there in terms of knowledge of background, we need to understand how to identify clinical interventions from these genetic tests. So there's CPIC guidelines to start. This is a very wide-scale consortium that involves several hundred providers, informaticists, pharmacists that help assist in interpreting our genetic tests. So what they do is they come up with multiple guidelines. At this point, we have 26 gene drug guidelines. And the importance of noting what CPIC does is that this is an interpretation, not an indication of when to test. So that is under the discretion of providers to do so, but this helps us figure out what kind of recommendations we can give our patients. And the levels of evidence that it categorizes is from a to D scale. So A or A/B indicates a strength of evidence moderate to high, and that also determines if we have a strong or likely prescribing change recommendation. So as you can see, there's a lot of recommendations where we're able to give a strong suggestion, which are actionable or targetable, but we also have at least half, if not slightly more, that only have a weak strength of evidence. And that's because with genomics being such an evolving field and so new, we don't have enough evidence to support that these variants are impacting the treatment efficacy of these patients. So to take a look through one of the examples, I inserted what the website looks like. So over here, we can take a look at one example in particular, which is therapurines and TPMT and NUTT15 mutations. So this highlights the guideline publication information on any updates to clinical recommendations for those genotypes. And it also provides any updates further since the guideline was published. It also indicates on the bottom any frequency tables or allele definition tables, so it can help you determine if this specific mutation is relevant for your patient population based on race or other factors that they're able to identify. And then below here, they also have gene resource mapping, drug resource mapping, and lastly, some clinical decision support. So what all of that means is it gives you a lot of resources to be able to identify, and if this link would have worked, I would have shown you how these guidelines show the map of the mechanisms behind these mutations and why they're impacting our drugs the way they are. So before going through our other resources, I wanted to walk through specific examples. So I hinted at it with the CPIC guidelines, but today I want to talk through mercaptopurine. So mercaptopurine is typically used to treat acute lymphoblastic leukemia, and that's commonly in the pediatric population. Off-label, it's also used for inflammatory bowel disease. So highlighted in gray are the enzymes that I would like to discuss, which is the TPMT that helps convert our mercaptopurine into inactive metabolites, but it's also affecting the efficacy, aka the RNA damage and DNA damage that subsequently impacts apoptosis. And most of us have a normal function of TPMT, but there's an estimate of around 14% that may be heterozygous and contain at least one TPMT allele lacking function. So that means that around 3 to 14% of us are immediate, intermediate metabolizers, and there's a smaller percent that have a lack of function in both. So 0.3%, most likely in European ancestry is when this occurs. And so typically we have recommendations to have alterations to our doses. If starting dose is already at the higher range, and higher is considered maybe 75 milligrams per meter squared, there is a recommendation to lower that starting dose in intermediate metabolizers and then have a significant dose reduction of around 10-fold if you're a poor metabolizer. So in order to find out more information about this type of drug metabolism, if it impacts our patients, is through some other resources that we can use in combination with CPIC guidelines, one of which is PharmGKB. The intended audience for this encompasses patients, clinicians, and researchers. So it's a really versatile resource and is publicly funded by the National Institute of Health, has a similar categorization of evidence, and it also links to clinical guidelines within CPIC as you're looking through the medication if you're searching in their website. So for instance, we have mercaptopurine here again, and you can see the pathways that I was hinting at before. So that same image that I showed you is also linked in their website. So you're able to look through and directly see the theoretical impact that your variant might have on the patient. And that's spelled out here further. You can also take a backwards approach. So if you suspect that the patient has a TPMT mutation, instead of looking up the drug, you can look up that variant. And you can see the drug label annotations here, as well as the level of actionability and recommendations for that specific variant. And here they have all of the variants listed out. And as a highlight, the TPMT1 is mainly utilized for the normal gene type, as they're not able to catalog it in any other way. So one is commonly a normal metabolizing individual, and then the rest have varying degrees of metabolism. And towards the right here, we can see pretty clearly, you can identify what might be the implication in patient care, of which notably dose reductions, as I've mentioned before, is a typical recommendation. There's also some concerns for adverse effects, such as ototoxicity, myelosuppression, and leukopenia, because the patient won't be able to clear the drug as readily. And that's for, this takes into account both mercaptopurine as well as other medications that might be impacted. So there's also, in case you want to delve into the information further, there's also literature that's specifically sourced here. So there's no need to do a deep PubMed search if you're trying to identify specifics behind what literature is available for TPMT variations. It's all listed out here clearly for us. Another interesting resource that we can utilize is OncoKB. So that has 685 genes cataloged in it, which amounts to over 5,600, 5,700 genetic alterations that it catalogs. And it also encompasses over 100 cancer types and 100 drugs. So within here, we see that there's a lot of medications that have FDA-approved drugs and genes that are associated with those drugs. So potentially targetable and actionable mutations over there. And then we get to the right side where we see some standard care, some clinical evidence behind the gene-drug associations, and then we see some level R1 and R2 full resistance with the gene linkage. So an example here, I wanted to highlight how the information is showcased, mainly because what they use is what's called a lollipop diagram. So that's really important for clinicians and us as pharmacists to be able to visualize where the variant is happening. And I appreciate these types of plots mainly because it's color coordinated. You're able to see specifically where the mutations are occurring based on what the specific mutation you're looking at within OncoKB is. And it also identifies the specific cancer types that might affect. So in this case of ALK mutations, we see that there's a significant amount of gene alterations in embryonal tumors, and then it drops down pretty significantly, but second and third are non-small cell lung cancers and salivary gland cancers. So we can take a look here and have, when you further look down on the ALK website within OncoKB, you can utilize the different headings. So we have annotated alterations, therapeutic, diagnostic, and FDA recognized content, all of which have a very parsed down view of what types of recommendations we can provide and what the mutation might be. So in the instance of ALK mutations, if the alteration is an amplification, they've identified that it's likely oncogenic in source, and the mutation effect is a gain of function. Now, we'll circle back to yet another publicly funded source, which is the FDA table of pharmacogenomic biomarkers. This is an identification of all medications within the FDA pharmacogenomic recommendations. So these are very heavily scrutinized. It's a great resource that predominantly utilizes drug labels and gives you a really good view of what the package insert recommendations may be, as it's directly from the source. This information is strictly from the FDA. So it provides information regarding the risk for adverse events, drug exposure and clinical response variability, genotype-specific dosing, mechanism of action, trial design, and as I mentioned, additional package insert information. So this is what the list looks like. We have a significant amount of drugs that is a scrollable text. A lot of this information is also exportable in an Excel spreadsheet. So what you're able to identify when you click into a medication is the specific products that are available on the market, and then all of the labeling information. So this is our main go-to source if you want any package insert information on your medication. Other resources, there's a vast amount of them, but the main ones I wanted to highlight include BioPortal, which is this large ontology of all these biomedical data resources. That's very useful if you want to see all the information that's out there. There's also ClinVar, which is a specific search engine that helps you identify like a simple Google search if you want to look into your genomic variations. And they'll also segment it under condition, gene, origin, whether it's germline or somatic, various citations within PubMed, because it's also associated with the National Institute of Health. And ClinGen is a subset that's also similar to our ClinVar search. So within UW Health, we have two resources that are widely available to us. One of them is the Precision Medicine Molecular Tumor Board, and that is in partnership with the UW Collaborative Genomics Core and the UW Carbon Cancer Center. They help identify any cases that are submitted when a patient is concerned for having some genomic variant, and they discuss all the risk benefits for targeting those specific genetic markers that they've identified. This is provided at no additional cost to the patient, and it's a very collaborative, great exercise in identifying which types of therapies are best for our patient We also have the Center for Human Genomic and Precision Medicine here. That was founded more recently in 2018 that promotes research, clinical and educational opportunities for precision medicine. And so the way that this is performed is typically when a patient is undiagnosed, they're seen in clinic, they then become enrolled in a study and they have their whole genome sequence performed. They also help, this specific center also helps with developing new diagnostics and predictive tools. And they're also heavily involved in research in wet labs and clinical trials to help improve genetic testing for our cancer patients. So I have our second assessment question. Which reference is most known for providing predominantly genomic information from the package insert? FDA, ClinVar, PharmGKB, or OncoKB? I saw a few responses. So the correct answer is FDA. So while some of the other resources might be able to identify some information pertaining to what would be identified in the package insert, the best way to find this information is through the FDA table, as that's straight from the horse's mouth. They're the ones who are assisting with making these recommendations on the package inserts. And so they're essentially the gold standard for finding this information. Now that we've talked about the background and how to incorporate that genetic information into guideline-specific clinical recommendations, how can we incorporate all this information we know into our patient records? And that is what I'm most excited about, because Epic itself, our personal EHR, has a genomics module. So what it does is it takes genomic data and it converts it into a more usable tool, which is within the EHR. So ideally, what we have is we have a pharmacogenomics module that utilizes all of this genomics data and attempts to standardize it. Now, I want to emphasize the word on attempt, because this is, as I'll walk through, a much trickier task than it may seem at first. What you're trying to do is you're trying to provide information about all these various genomic variants into the patient chart, which is not well standardized at this point and has a lot of data behind it. So it requires a lot of server space, requires a lot of upfront work, but the rewards, hopefully, if in the next coming years, can get parsed down into a better format, is very invaluable. So ways that we can utilize this is basic analysis of patient populations. An example, utilizing slicer dicer to try and determine trends in our patient population. Clinical decision support, so utilizing best practice alerts, other things that might populate in the background of our health records that can utilize these evidence-based suggestions from CPIC guidelines and other resources. We can also utilize this pharmacogenomics module for phenotype documentation, and that also helps with interpretation of data. And lastly, communication with our fellow providers. So having a specific module that has this genetic information available in an electronic format also lets us more readily able to send it to our fellow partners. How does this specific genomic indicator work is the main question. So these genomic indicator records can be added by geneticists or clinicians to the what's called genomic indicators activity, and that unfortunately is currently still a manual process. So they have to hand insert all of the genetic information into the chart. But essentially what happens is the provider orders a genetic test for a patient, then the lab processes that genetic test, and then those genetic tests are entered into Epic, and then there is a variant stored on the patient's chart. And then finally, this genomic indicator is created on the patient chart. To walk through yet another example, this is taken from the CPIC guidelines. So this shows you how the information is organized in a table format for CPIC recommendations for warfarin variants. And so what ends up happening is we utilize that information from the CPIC guidelines to determine specific dosing algorithms. And in the background, there's a lot of complexity behind making an actionable intervention from those CPIC guidelines as a result of the pharmacogenetics dosing recommendations. So there's a lot of calculations that the computer has to evaluate in order to reach a single objective warfarin dose. And that's why for now we have to utilize a lot of clinical judgment, because these equations become very complex, as you can see. There's a lot of factors that are involved with a lot of constants here that have been validated by a lot of pooled population data, but it's still very hard for us to be able to utilize that and have an impact within our EHR. And so that's why instead of dosing recommendations, a lot of institutions have focused on utilizing best practice alerts. So that applies evidence-based medicine that we've identified within our guidelines and within all of our resources to flag specific medication interventions that we can make based on the patient's data. Ideally, these are actionable BPAs. What that means is that's a best practice alert that has a specific target in mind, whether that be consider lowering a dose or consider removing a medication. Those interventions specifically should be spelled out in a perfect world within that alert so that the clinician knows how to follow up on that information. An example of how this gets stratified is also from CPIC guidelines. This was identified for 6-mercaptopurine, whether or not it has these two variants or mutations. And this is what goes on in the background in order for the best practice alert to fire. So it has to identify if a TPMT result is on file, if the other mutation that I didn't discuss, which is the NUDT 15, is available, and then it can finally reach a conclusion of whether or not it should fire an alert for the clinician. Some of these alerts can show up, I'm sure we've seen these in yellow. These are moderate or a lower sensitivity to phenytoin. So an example, this shows a CYP2C19 intermediate metabolizer, and this is taken from an outside institution, so this is not utilized currently at UW Health, but you can see the recommendation in their case is to remove the order based on the genetic information that's available on the patient's chart. And then there's also a critical alert that shows if there's a safety issue involved, and this shows that there's an increased risk of myelotoxicity as a result of what was flagged in the chart. So these are two examples of best practice alerts that might look similar to ones that we have on board, but these are ones that specifically highlight genetic variations and targetable clinical interventions as a result of that. In our current state, We have information scattered across different tabs, so we don't have this within a specific structure. We have this information in pathology, media, labs, and letters, not in an electronic format. It's usually available in PDF files. And there's a filter technique that we can utilize to minimize the manual search, but there's currently no protocols in place for any inpatient or outpatient genetic panels outside of the oncology realm. So this mainly applies to our oncology patients, but there's certainly areas of opportunity within neurology, cardiology, psychiatry, and any other places that might have drugs that impact their care in relation to genetic variants that might impact their metabolism or other factors. So this filter technique I wanted to highlight on, as this can be applied if you ever work with oncology patients. So what you do is go into chart review and utilize the media tab to filter out the HC reference lab reports, lab managed results scan, and lab results scan hyphen external, as these are the main sources that populate the genetic information. So this is an imperfect workaround that we found can help us find this information faster as opposed to trying to sift through all the information that might be located within media. There's certainly a lot of barriers to pharmacogenomics testing currently. Reimbursement is still an issue and is dependent on insurance. Sharing information is also limited as genetic information may impact offspring, and we still don't have a good protocol on how to identify this genetic information to the offspring. So unless the clinician has a relationship that was previously established, they're not aware of these genetic test results. So there's a lot of ethical considerations as a result of this. And there's certainly no neat way to fit all of this information into the health record currently. Some future implications into utilizing pharmacogenomics data in the electronic health record is this pharmacogenomics module that will become live at UW Health this coming April. So initially, this is going to be using Strata data and then incorporating Tempus, another pharmacogenomics company that will be providing all of the information in a readily electronic format as opposed to the PDFs that we've been receiving. There's also the potential to utilize cloud storage in the future, and this we might see in the next coming years or decades, but that would eliminate the use of servers and the challenges that we have with data storage. We also have the options to utilize haplotype and diplotype documentation and parse down the information even further. And there's also potential to integrate with ClinVar in the future to where we can see all the most current guidelines and genetic variation information. So this leads us to our final assessment question. What companies are available for pharmacogenomic testing through electronic health record pharmacogenomics module? Strata, Tempus, Invitae, or A and B? So I see a lot of D. So luckily we're getting this pharmacogenomics module in April, so you'll see a lot of the Strata and Tempus information populated as a result of that. So here are my references, and that should be the conclusion of my grill. I'll take any questions at this time. Okay, if there's no other questions, hopefully you're able to apply this in the future when we utilize this in our oncology patients, and hopefully this spreads to other patients as we get more information on genomic variants in the future and targetable interventions.